All right, well, it's Thursday again, so welcome back to Science in the Age of COVID-19. Hope everyone's staying safe and sane. Today we are super excited to have Dr. Tim Sheahan from University of North Carolina Department of Epidemiology. Tim did his PhD in the lab of Ralph Barrick at UNC working on SARS-1. One of those names that now almost everyone in the world is aware of and following closely. He did his postdoc in the lab of Charlie Rice at Rockefeller, studying hepatitis C, where he met our very own Kim Ratola. And then after a brief stint at GSK, learning high throughput screening, he ended up back at UNC working on emerging viruses. So today he's going to tell us, hopefully some ways that we can keep this one under control, and then how we can be better prepared for the next time this happens. So, Tim?
Hi. Can everybody hear me?
That's great, dude.
Okay. Just going to share my screen. Perfect. Does this look okay?
Looks great, man.
Okay. So here we go. Thank you for the introduction. I usually show this slide to kind of tell people how I got to where I am today, but you did such a nice introduction. I'm going to just touch upon this briefly because I never really thought that I would be where I am today. And in fact, like all through undergrad, basically high school, undergrad, and even working as a tech in Boston before grad school, I really was focused on being a singer in a punk rock band. I'm on the right with a lot of hair that I don't have anymore. But during this time, I really began to appreciate the kind of the same things that go into writing songs and performing songs kind of go into designing experiments and communicating science. So I went to grad school at UNC, postdoc at Rockefeller, brief stint at GSK, and I've been at UNC since 2015, once again working on coronavirus. So my research kind of falls into three bins where we are studying the emergence potential of coronavirus. So trying to figure out what viruses have epidemic potential out in zoonotic reservoirs. Kind of related to that, we're focused on developing broad spectrum therapeutics for times such as these. And also we're trying to figure out how human genetic diversity affects the outcome of viral infections. But today I really want to focus on emerging viruses. And newly emerging viruses are new in the sense that they're not known to have previously infected or caused disease in humans. So they're not new to Earth, they're just new to us as humans. And this is a cartoon just showing kind of how this works. So lots of viruses circulate among wild and domestic animals. Through a spillover event, sometimes these viruses enter humans. If they're capable of replicating in people and capable of transmitting from person to person, you could have a localized outbreak or a localized epidemic. But because it's so easy to travel throughout the globe, one sick person getting on a plane can send this virus throughout the Earth, causing a global epidemic, or what we're seeing now is a global pandemic. And emerging viruses are a global problem. This is a map adapted from a Marston and Fauci paper from 2014, just showing some newly emerging or re-emerging viruses and where they popped up. And one thing I just want to note about this slide is that most of the viruses on this slide have RNA for their genome. And that's important because as RNA viruses replicate, they make mistakes in their genome. So what I'm showing you here is just genome size on the x-axis and the mutation rate on the y-axis. And as you can see in the top right corner here, RNA viruses have relatively small genomes compared to us over here, but they make many more mistakes as they replicate. And coronavirus kind of live, even though coronavirus, they are RNA viruses, they kind of live in this space, and I'll tell you why in just a little bit. So coronaviruses, this is an EM taken by Fred Murphy of SARS-1. Coronaviruses get their name from the Latin word for crown, which is corona, due to the spike-like projections that kind of come out of the virus particle, which resemble the spike-like projections on a queen's crown or a king's crown. And coronaviruses are the largest RNA viruses. So this is just showing lots of different RNA virus families, and then the genome size and the x-axis. And as you can see in red, we see coronavirus have genomes around 25 to 30 plus kilobases. And the reason that they're able to maintain these large RNA genomes as compared to these other RNA virus families is due to the existence of a proofreading enzyme called XON, which just basically comes from the word exoribonuclease. And this was work done by Mark Dennison's lab showing that XON really drives proofreading during coronavirus replication. And what I'm showing you here is a neighbor joining tree created from the spike glycoprotein. So this is a protein tree. And as you can see here, if you look at the comparison versus SARS here in this heat map, within a subgroup or a branch, so this is 2B where SARS-1 and SARS-2 reside, there's high identity of this protein. But once you step outside of this branch, you see this precipitous drop in amino acid identity. So comparing SARS to MERS, they're not that similar. But luckily, a lot of the enzymes that are required for replication, this one is a really important enzyme called the RNA-dependent RNA polymerase, which guides RNA synthesis. There's much more similarity among the family as well as within each branch. So they're basically 100% identical when you look within the 2B branch here that SARS-1 and SARS-2 live in. And perhaps more importantly, if you're interested in developing drugs that target the RNA-dependent RNA polymerase, the pore that the nascent RNA is created within is highly conserved. So this is just, we're modeling the family-wide diversity in the RDRP onto the structure of the RDRP, and you can see the dark purple kind of pore here, so it's highly conserved in the pore. What I'm showing you here is a picture from my virology textbook from 2001 from grad school. And before 2002, coronaviruses weren't really known to cause severe human disease. And I've highlighted the two known human coronaviruses at this time in this table, 2290 and OC43, which cause the common cold. And most of the other viruses that fill up this table are of agricultural or domestic animal importance. And that all changed with the emergence of the original SARS coronavirus in 2002, 2003 in Guangdong province, China. It's believed to have emerged from the bat reservoir species through a civet intermediate host, ultimately causing 8,000 cases with about a 10% mortality rate in 26 countries. And this epidemic was brought under control by classic public health measures. And with the emergence of the original SARS coronavirus, it really created a new paradigm for human coronavirology, in that we have a newly emerging human coronavirus that causes severe disease in humans, and the fact that this virus was carried rapidly throughout the earth by air travel of infected people. And this new paradigm for human coronavirology was really solidified with the discovery of MERS coronavirus in 2012, like SARS-1. It's believed to have come from a bat reservoir, but this time we're having transmission to humans through a camel intermediate host. And MERS is endemic in camel populations in the Middle East and continues to see human cases to this day. So this is an epi curve from the WHO, just showing the numbers of MERS cases over time. And the red peak that you see in the middle was a small outbreak in South Korea in 2015, where a traveler got close to a camel, acquired MERS, and brought that infection back home. It ended up essentially causing a nosocomial outbreak of MERS coronavirus. And so, bats are really important reservoirs for coronavirus. And coronavirus are found in bats from all over the world. These are just some publications showing that you can find coronavirus in bats in Central America, Africa, Asia, Australia, and the United States. So all of this basically is leading to the notion that coronavirus as a family have emergence potential. And it's believed that all or almost all known human coronaviruses originally emerged as zoonoses. And there's no reason to believe that this won't continue happening in the future. So there really is a need for coronavirus therapeutics since 5 novel coronaviruses have emerged in the past 20 or so years. And this is not a uniquely human issue. There have been 2 newly emerging pig coronaviruses as well, PDD and SADS coronavirus. So this is really just an aspect of coronavirus biology that we need to think about and address. So what we're trying to do to address this is develop broad spectrum therapeutics. And the more traditional approach would be to develop one drug for one bug. So we have MERS coronavirus. Let's develop an exquisitely sensitive and specific drug for MERS. But I think that has limited utility given that we're dealing with coronavirus, which have this emergence potential. So rather than do that, we want to make one drug for many bugs. So we want to develop broad spectrum therapeutics for coronaviruses that we know about today. But in addition, we want to develop therapeutics for those that might emerge in the future. And we've been doing this work since maybe 2014, 2015. Through public-private partnerships with Gilead and academic collaborations as well, with the goal of developing some kind of therapeutic that has family-wide antiviral activity. And this is just a picture of the 2 kind of academic institutions leading the charge here. We've got the UNC Chapel Hill team, which is headed up by Ralph Barrick and myself. And then we have a Vanderbilt team headed up by Mark Dennison on the right. Mark and Ralph have been collaborating on coronavirus research for decades. So it's really good to see this collaboration continuing to flourish. So just one note about the working environment that we have. And these viruses are dangerous human pathogens, and they have to be worked with safely. So you have me working at biosafety level 2, wearing my safety glasses, which I wear all the time, lab coat and gloves. I'm working at biosafety cabinet, of course. And then you have my friend Amy Sims, who's wearing a full Tyvek suit. She's got a powered air purifying respirator on her belt, feeding sterile air into her hood as she does her work. So, all biosafety level 3 labs are not created equally. So if you're used to working on bloodborne pathogens like HIV, the PPE and the work practices in that environment would be different than working on a respiratory pathogen. So at UNC, we've got lots of really interesting and powerful tools to do this work, from the molecular virology, which allow us to construct recombinant viruses, primary cells to look at antiviral activity in vitro, as well as cell lines, and really good models of in vivo pathogenesis. And the kind of drug that I want to talk about today are nucleoside analogs, which basically mimic our building blocks of life. And the 2 that I'm going to focus on today are shown on the right, remdesivir and EIDD 2801. So remdesivir was originally being pushed as an Ebola antiviral, and it had shown in vivo efficacy in monkey models of Ebola virus hemorrhagic fever. It was given to thousands of patients in West Africa in the most recent Ebola outbreak, and has since then been given to thousands of COVID-19 patients as well. And we wanted to, originally when we started this collaboration with Gilead and Vanderbilt, we wanted to look in this model to look to see if we could get antiviral activity against the original SARS and MERS. And this is a primary human airway epithelial cell culture system that recapitulates aspects of the biology in our conducting airway. So these cultures are differentiated at an air-liquid interface. The apical surface of the culture faces is exposed to the air, and the basolateral surface is under some kind of growth medium. These cultures have ciliated epithelial cells on the apical surface, which actually beat in a coordinated fashion, and goblet cells that secrete mucus. So you actually have to like rinse the snot off of the apical surface of these cultures every few days as it builds up. And they're also really good models for to study coronavirus, virus and host interactions and replication. So this is what we did. We set up some antiviral assays for MERS coronavirus on the left and the original SARS coronavirus on the right. And this is work that was published in 2017, but work that we really started doing maybe around 2015. And as you can see, we see a dose-dependent reduction in infectious virus production, which is measured on the y-axis, with increasing doses of remdesivir for both SARS and MERS coronavirus, with sub-micromolar EC50. So EC50 is just a measure of potency. So the lower the number, the better. So then we wanted to really get a feel for the breadth of this activity. And was this a broad spectrum antiviral for all coronavirus? And for that, we leaned on the reverse genetic systems that have been built up by the Barrick lab, beginning in the early 2000s, and just all the papers for all the reverse genetic systems are shown in the bottom right of the slide. But basically, you have a really big RNA genome, and we basically split that up over multiple fragments of cDNA, which are coded into plasmids. So you can manipulate each fragment separately. If you want to just alter one gene, you only have to like do the cloning for with one of these fragments. And then when you want to assemble a recombinant virus, you basically cut out the viral cDNA, you ligate it together because the ends have specific restriction enzymes that allow for directional ligation. And you make in vitro transcribed RNA from this DNA template, and then all of a sudden you have recombinant genomic viral RNA, which you will electroporate into a susceptible cell like a vero cell, and out comes recombinant virus. So with this system, we're able to build any kind of coronavirus. So if someone isolates a coronavirus from a bat in Africa or in Asia, and they publish that sequence, we can recreate that virus in the lab in North Carolina and study its biology. So using these techniques, we are able to measure antiviral efficacy in primary human airway cells and other cells to an array of different coronaviruses shown here in this neighbor joining tree. And the color of the text basically relates to the animal within which you find this virus. So PD-CoV is a newly emerging porcine coronavirus. And we can see that remdesivir has potent antiviral activity against all these different coronaviruses, which recapitulate almost family-wide genetic diversity. And I'm just showing you some of that data on the right against human coronavirus NL63. So this is a common cold-causing coronavirus. HKU5 is a MERS-like 2C coronavirus. HKU3 is a SARS-like coronavirus. And then SHCO14 and WIV1, these are interesting because these are bat coronaviruses, but they're able to readily infect human cells without any adaptation. So basically, right out of a bat, these viruses are poised for emergence into humans. So the take home from this is remdesivir works in vitro against all these different coronaviruses, including those that have the potential to emerge in the future. So now I want to kind of jump into the in vivo work that we've been doing. And one thing to note is that mice are not human. And as such, the pathogenesis models for coronavirus are very different in a mouse than a human. So I have kind of a general scheme of how like SARS or MERS pathogenesis might go in a person, where you have ramp up of virus replication in the respiratory tract. It peaks 7 to 10 days after the onset of symptoms, and goes down after that as your adaptive immunity revs into high gear. So in a mouse, disease is much more compressed. So the whole thing plays out over a week. So unlike in a person where you have peak virus replicating in the airway, 7 to 10 days after the onset of symptoms, in a mouse, it's day 1, day 2 post-infection, virus peaks, and it goes down after that. And depending on the age of the mouse or their genetic background, this infection will result in survival or mortality. So we set up the initial studies for remdesivir in a SARS-1 model some time ago. And one thing to note is this right here. To look at remdesivir antiviral activity, you have to do it in a specific knockout mouse that is knocked out for carboxyl esterase 1C. And this is a serum esterase that is present in mice that is not present in humans. So if you put remdesivir, if you expose it to like a wild type, from serum from a wild type mouse, the stability in the half-life is really short. And so to better model the pharmacokinetic properties of remdesivir that you see in a person, we do all of our mouse work in CES1 knockout mice. So we set up this following study where we have prophylactic treatment beginning day 1 before infection, or therapeutic treatment day 1 after infection. And we're measuring weight loss and pulmonary function over time. And then we looked on day 4 to see if there was a change in virus titer, so our virological endpoint. And just to note, remdesivir is given twice a day subcutaneously. So what we saw was that therapeutic remdesivir and prophylactic remdesivir could diminish weight loss associated with SARS-1 infection. And just one thing that I did not mention is this is a mouse-adapted SARS-1. The terminal time point here, day 4, is just blown out in the center graph here. So you can see the difference between vehicle and treated, prophylactic, and vehicle and treated therapeutic. Perhaps more importantly, we see a significant reduction in viral load in both prophylactic and therapeutically treated mice. And one really cool tool that we have in the Biosafety Level 3 lab is we have the ability to measure whole body plasmography. And from this, we get insight into pulmonary function in live mice. So you put mice in these plastic chambers, which live in a biosafety cabinet. And after an acclimation time, the machine will measure the force of each breath, the duration of each breath, the inspiration, the expiration. And from this, you get lots of different metrics which tell you something about how pulmonary function is happening in the mouse. And so what we found in this model is that therapeutic remdesivir can improve pulmonary function. So the metric I'm showing you is called PENH, which is a kind of measure of pulmonary obstruction. And it's kind of depicted in this little cartoon on the bottom right of the slide. A high PENH is bad. That means loss of pulmonary function. So with prophylactic treatment shown in red or therapeutic treatment shown in orange, we see a reduction in the loss of pulmonary function. So this was good. So then, because SARS-1 is basically an extinct virus, we wanted to look at the time at a virus that had the potential to become, to cause global epidemics, and that would be MERS at the time. So then we set up the following study to look at in vivo efficacy in MERS model of viral pathogenesis. And another wrinkle in the MERS story is that MERS coronavirus does not infect wild type mice. So the Barrick lab humanized the murine ortholog of the entry receptor, which is called dipeptidylpeptidase 4 or DPP4. So they humanized the murine ortholog with CRISPR, resulting in a mouse that could be readily infected by clinical isolate, but required passage. So the virus had to be passaged in mice to develop severe disease that was reminiscent of that seen in people. So we set up this study, which is similar to the SARS-CoV study that I mentioned a minute ago, but we added another few treatment arms due to a clinical trial that was underway in Saudi Arabia, where they were treating MERS patients with lopinavir-ritonavir, which is an HIV protease inhibitor cocktail. So they were giving lopinavir-ritonavir and interferon beta to see if they could treat people in the hospital with MERS infection. So because this was currently being evaluated in people, we thought we would look to see what effect that would have in mice. So if you look at virus lung titer in this model first, you can see remdesivir can reduce MERS lung titers, and lopinavir, ritonavir, interferon beta didn't really have any effect. And data I'm not really showing you here is the in vitro data showing that lopinavir does have an antiviral effect against MERS, but at very high concentrations that are really hard to achieve in a person. So when we set up the mouse study, we dosed them with a human equivalent dose. So it's important to note. So in collaboration with a veterinary pathologist that actually went to grad school with Dr. Kim Ratola and myself at UNC, we did this really cool study from mouse lung sections from this, the MERS drug study. And so what Stephanie did was she blindly scored these mouse lung sections shown on the right for the pathological features of ARDS. So ARDS stands for acute respiratory distress syndrome. It is an end-stage lung disease that SARS-1, SARS-2, and MERS patients sometimes find themselves with. So people who are in ICU on ventilators are often suffering from ARDS. So a few years ago, the American Thoracic Society published a scoring tool that we use to quantitate ARDS in these mice. And as you can see on the left, remdesivir was really the only treatment that significantly diminished the pathological features of ARDS. And you know, people always ask, you know, what about the development of antiviral resistance, since this is obviously a problem with antiviral drugs, especially with antiviral drugs against RNA viruses, which, as I mentioned before, have this inherent plasticity in their ability to change and evolve when under pressure. And this is work done by Mark Denison's lab, where they used a model alpha coronavirus called mouse hepatitis virus, and they passaged MHV in the presence of remdesivir 23 times. And only then did they see a few mutations pop up, and they popped up in the RNA-dependent RNA polymerase, which is not surprising. So this said to us that there's a high barrier of resistance. And with these mutations, when you put these back into the infectious clone for MHV, or even if you transfer these similar mutations to SARS-1, the resistance that's gained by these two mutations is only results in about a 5-fold shift in the IC50. So it's not like some of the resistance mutations that you see crop up for HIV, where the virus basically can replicate you know, pretty normally in the face of antiviral treatment. But importantly, one thing that we found which was really cool, which if you take a remdesivir-resistant virus and you treat it with another nucleoside analog, the one I'm showing you on the right is called NHC, which I'll talk about in a little bit, you see that the remdesivir-resistant viruses are more susceptible to the antiviral activity of this other drug, basically saying that remdesivir resistance mutations do not transfer resistance to this other related but different nucleoside analog. So that's what you're seeing here. We have fold change in titer on the y-axis and increasing doses of NHC. And the doubly resistant virus here is shown in red. You can see the difference between wild type and the resistant virus. So that basically you're dropping titer more in the resistant virus. So then we wanted to figure out very recently, you know, will remdesivir work against SARS coronavirus 2? Because we've shown in vitro activity and in vivo activity against SARS-1 and MERS coronavirus. So we were very interested upon when we found out that the newly emerging virus in China was a coronavirus. We wanted to figure out would remdesivir be helpful in treating what would eventually become called which would eventually be called COVID-19. So we set up studies in human airway epithelial cell cultures. On the left-hand side, it's basically an antiviral assay looking at virus production with increasing doses of remdesivir. And we see that in 2 different studies, we see a dose-dependent reduction in SARS-2 virus production with increasing dose of remdesivir. And these data on the left, plotted as an IC50 curve on the right, you can see that the EC or IC50 values are pretty good, and they're comparable to that which we saw in SARS-1 and MERS. So as I alluded to before, we kind of had this conundrum with SARS-2, where if we wanted to look at in vivo efficacy, how would we do this when wild-type SARS-CoV-2 does not infect a wild-type mouse? So the first thing we thought of doing is just making a chimeric virus between SARS-1 and SARS-2. So we basically have reverse genetic system for SARS-1, and we replaced the RNA-dependent RNA polymerase of SARS-1 with that of SARS-2, because remdesivir is targeting this protein. And so we thought this might be a good surrogate while we work on developing better animal models for SARS-2. So that's what we did. And we performed some antiviral, some therapeutic efficacy studies, similar to what we did for SARS-1 and MERS, where we had CES1 knockout mice, and we treated them with remdesivir 1 day post-infection, or we treated them with vehicle. And what I'm showing you on the left-hand side is pulmonary function. So this is the 10H metric again. You can see remdesivir is able to prevent the loss of pulmonary function that you see with the vehicle. And importantly, we see a dramatic reduction in viral load in the lungs on 5 days post-infection. So that was good. So I kind of want to switch gears a little bit and talk about another nucleoside analog that we've been working on, kind of very briefly, because the data that we've developed for this drug kind of mirrors that that we developed for remdesivir. So this is EIDD2801 is a prodrug of NHC, the compound that I showed a few slides ago. So it's a nucleoside analog, and like remdesivir, it has broad activity against an array of RNA viruses, including flu, Ebola, DVE. And like remdesivir, we found that EIDD2801, or NHC, is effective against many coronavirus in vitro, and we showed therapeutic, prophylactic and therapeutic efficacy in both SARS-1 and MERS models. And this preclinical data has positioned EIDD2801 for deployment in humans with COVID-19. It has an advantage over remdesivir, which is an IV drug, because this is an oral drug, and so it could be used more widely and out in the community, which is hard to do for an IV drug. And this is now in phase 1 testing in humans, and if it's safe, this will move to phase 2 efficacy testing in COVID-19 patients. So we're really excited about seeing that. So now I want to spend the last few slides just talking about some of the new tools that we've been developing in the lab to better evaluate vaccines, antibodies, and therapeutics against SARS coronavirus 2. But I need to take a drink of coffee, so just give me 1 sec. Perfect. So about a month ago, we finished developing a reverse genetic system for SARS-CoV-2. And with that, it gives us the ability to manipulate the SARS-CoV-2 genome and do things with it. And so one of the things that we thought to do, since this virus does not replicate in mice, was can we remodel the spike and receptor binding interface such that we can create a more efficient interaction between the murine ortholog of the human receptor. So SARS-CoV-2 and SARS-CoV-1 uses angiotensin converting enzyme 2, or ACE2, as an entry receptor. And the murine ortholog of this receptor does not support SARS-CoV-2 infection. So using the reverse genetic system, we made 2 point mutations to remodel this binding interface based on structural modeling that a really talented grad student in the Barrick lab named Kenny Dinning did. And so Kenny made this recombinant virus, recovered this recombinant virus, and then transfected in either human ACE2 or mouse ACE2 into non-susceptible cells. And now you see, unlike wild-type virus, which does not infect mouse ACE2 expressing cells, now we see infection suggesting that, the remodeling of the spike receptor binding domain now facilitates infection through the murine ortholog of the human receptor. So Kenny, with help from lots of people in the lab, set up a series of pathogenesis studies with this mouse-adapted virus, which they're calling SARS-CoV-2-MA for mouse-adapted. So I'm showing young adult mice in the top, so these are 10-week-old mice, and aged mice, which are a year or more old. These are mice in the BALB-C background. If you infect them with the mouse-adapted virus, you see high levels of replication in the lungs of both young and aged mice. If you infect them with wild-type virus, basically it's undetectable. In young mice, there's kind of a blip of virus in the aged mice on day 2, but that's basically gone by day 4. So this was good. We had a virus that could infect a normal kind of laboratory mouse. And when we looked at pulmonary function in the young and aged mice, we could see an age-related increase in pathogenesis. So we see a greater loss of pulmonary function as measured by this 10H metric again in the aged mice. So we see a greater loss of function in aged mice as compared to the young adult mice. And when I look at these data, it kind of mirrors that which we see in people, where older people get more severe disease than younger people. And it's really interesting that both SARS-CoV-1 and MERS have a similar phenotype in the young and aged mice. So we're trying to model this age-related increase in pathogenesis that's seen in people for all of these emerging coronaviruses. So then we wanted to try and use this model for therapeutic evaluation. And back from my HCV days, I knew this guy named Jeffrey Glenn. He's a hepatologist at Stanford. And I've been in contact with him for some time, trying to get a coronavirus project with him going. And it's kind of never worked out until now. So I'm really happy to see this happening. And so he's been developing pegylated interferon lambda as a treatment for hepatitis delta virus infection. And he thought, why don't we try and see if interferon lambda can be used as a therapeutic to treat SARS coronavirus 2 infections. So we first looked to see if pegylated interferon lambda would exert an antiviral effect against SARS-CoV-2 in human airway epithelial cell cultures. So how this molecule works, basically, like type 1 interferon, it binds to receptors on the surface of cells, and it basically induces the activation of a transcriptional program, which in itself is antiviral. So it makes lots of interferon-stimulated genes, which have various antiviral activities. So when you treat HAE with different doses or diminishing doses of pegylated interferon lambda, we see a dose-dependent reduction in infectious virus production in these cultures, and we use remdesivir here as a positive control. So then, now, after we saw this in vitro effect, which was quite potent, we thought, why don't we look in this new mouse model and see if we can see an effect in vivo. So we set up a prophylactic and therapeutic interferon lambda study in BALB-C mice, where we gave a single injection, 2 micrograms, of pegylated interferon lambda, either 18 hours before infection or 12 hours after. And we saw with both of these, both prophylactic and therapeutic, a significant reduction in viral load 2 days post-infection. So basically saying that in a mouse, if you give them interferon lambda, the antiviral program resultant from that treatment is able to reduce virus replication of SARS-CoV-2. So that was really encouraging. And we're continuing to kind of push these models, the pathogenesis models, to make them more reflective of what's seen in people. So, you know, I think there are some challenges in treating acute viral respiratory infections. And it's kind of outlined in this slide where, you know, the classic is influenza. And I have just some example direct-acting antivirals, Baloxavir, which is a new antiviral. It's an endonuclease inhibitor, and then the Tamiflu, the neuraminidase inhibitor. So, you know, when looking at antiviral activity or efficacy in human clinical trials, basically you have to give these within 48 hours after the onset of symptoms to really have a profound effect on viral load and the duration of symptomology. So, you know, and that's very different as compared to something like hepatitis C, which causes a chronic liver infection, where you're basically giving a direct-acting antiviral to someone who has a sustained viral load in their liver, and you're just looking for disappearance of viral genomes in their blood. So this seems, at least to me, seems like an easier thing to attack just by the nature of the disease that it's causing. So when looking at, you know, treating COVID-19 patients with direct-acting antivirals, I think we need to really obviously look to other respiratory pathogens as we look to evaluate these things in the human clinical trial. And, you know, using a direct-acting antiviral in a patient who's had COVID-19 for 2 weeks, and they're only initiating treatment in this person 2 weeks after the onset of symptoms when they're on a ventilator in the ICU, you know, maybe a direct-acting antiviral isn't really the best thing for that patient at that time. So it's definitely complicated, and we'll see what the many trials that are happening throughout the world show in the coming months. So yeah, so basically I hope that I've convinced you that coronavirus as a family have emergence potential. And one way to kind of not only address the current COVID-19 pandemic, but also look to the COVID-20 and the COVID-22 pandemic, what do we do to better protect ourselves against emerging coronavirus in the future? And I think one of the things we can do is by developing broad-spectrum antivirals, vaccines, and antibodies that work against the viruses that we know about today, but also against those that might emerge in the future. And one thing that I think we should really be focusing on is not only developing really potent direct-acting antivirals, but I think we need to be coupling those with anti-inflammatories or molecules that might accelerate repair of the damaged lung to more holistically treat the disease, not just attack the virus, but more holistically treat the disease and perhaps extend the window within which we can treat and have an improved outcome. So like the Tamiflu situation, it would be better if we had a bigger window than the first 48 hours after the onset of symptoms to give someone a drug and improve their outcomes. So this is my last slide. I'd like to thank everybody at UNC in my lab and in the Barrick lab, our collaborators in the Swanstrom lab who did a lot of cool sequencing for EIDD 2801, which I didn't really have time to talk about today. Our collaborators at Vanderbilt in the Denison lab, folks at Emory, everybody at Gilead, and our funding sources at NIAID. And I will stop sharing and take any questions.
Excellent, Tim. Thank you very much. Amassing the list of questions here. I've got a bunch that I may go ahead and start with. So yeah, you showed us a lot about remdesivir and how promising it looked in the animal models. Um, you may have a slightly different take on this, but my opinion is that it's looked pretty disappointing in humans. The New England Journal of Medicine study, I mean, that was an embarrassing mess, no control group, cherry picked data, and even then the outcomes weren't great. The leaked China study showed no effect. And then only the recent NIH study, was somewhat hopeful, but even then they changed the clinical endpoint 3 weeks before the study was done. And as of this morning, they still haven't released the full data set, which has everyone wondering what it is that's in there that they don't want seen.
Right.
So what's your take on, how do you think it's going to work in humans?
Yeah. You bring up lots of good points. I think, what I know is what Fauci said in that one press conference where you had a decrease in the time people were in the hospital with treatment, and they basically didn't talk about any virological endpoints. And I think it's like a pretty high bar to be doing clinical studies in hospitalized patients who may be fairly progressed into their disease process in the course of their disease. So, and that's kind of why I was showing the Tamiflu versus HCV versus COVID slide to highlight that. The Tamiflu and Baloxavir study was an outpatient study. So these are people who are within a couple days after the onset of symptoms. So I think for remdesivir to have the best effect, that's the kind of study that needs to be done. And the fact that it's an IV drug makes that fairly complicated. So I don't know, I think, it's definitely not jaw-dropping, the data that I've seen, as you mentioned, but I think it has the potential to look better, but maybe in a different kind of trial design.
Well, the first trial, the New England Journal of Medicine one, it actually, one of the reasons that it got hammered was that they seem to have included, people that weren't really sick. So their definition of ill was 94% or less saturation.
Oh.
So, in the first study, those people, there was no one in there that was really sick.
Right.
And even then there were, ill-defined participant dropouts that, a little disconcerting.
Yeah.
And then, not to pile on to remdesivir, but, as you brought up, if it needs, a long intravenous infusion, if I get sick and I'm in the hospital getting an intravenous infusion, I'm probably going to ask for convalescent plasma or an IL-6 receptor antagonist like tocilizumab instead of remdesivir. That was the case with its failure in Ebola. It was not that, it's not that it had no efficacy, it did, but it was, if someone was getting infused, there were actually, better options, which is why it fell out of favor for Ebola.
Yeah, no, I completely agree. But, it is potentially proof that you can treat a severe coronavirus infection with an antiviral drug. And, it's something that can be built upon. So I think, it's disappointing maybe, but encouraging at the same time.
Yeah.
And I'm sure that, people can do better.
Yeah, so I'll switch to my second question, which is, hopefully something I'm super optimistic about is, 2801, if that works well, I mean, that could be a game changer because, you know, if that's a pill, you know, that we're all picking up at CVS, then, you know, we could really bend the curve that way a lot
Yeah,
without getting too much exposure.
right. And you know that brings up a good point. You know, you're um, ideally you're not just treating hospitalized patients with a direct acting antiviral. Like it would be better, you know, if there was a potent antiviral, it would be better to get that out of the community and prevent people from coming into the hospital, prevent people from transmitting to the people in their household. Like that would be obviously a much better situation. So we'll see how that pans out.
Um, uh, can you tell us more about, you said it's in phase one. Do you, what partner are you guys using for that?
So yeah, so that drug is was originally developed by a group out at Emory,
Right, right.
and they licensed that to a place called Ridgeback Biopharma.
Okay.
So they're dealing with the clinical development. So like one important thing to note is that we played no role in the creation of these molecules, and we are also playing no role in the clinical development of these molecules. We simply just provided the preclinical, like proof of principle data to enable the human clinical trial. Um, so, you know, I like most of the people who are here deal in a lot of rumorology. So, like the rumors that I've heard are, you know, that it's looking good in phase one safety,
Oh,
and
fantastic.
we'll see what happens in phase two.
Yeah. Okay. Yeah, that's no pressure, but we're we're all waiting for that. Let's see. I wanted to change gears about uh, development of more things coming out of the jungle. So you know, obviously, SARS-2 came out. Since you know, you showed that picture from your textbook that only had 229E and OC43. And then, you know, you showed us the two pigwins. You know, now we also have NL63 and HK1
Yeah.
circulating. Where did those come from? And, you know, are we going to get more and more common cold coronaviruses in us at the same time that we're getting all these bad ones as well?
That's a really interesting point. So, you know, one thing that with the emergence of SARS-1, which kind of occurred during the explosion of sequencing technology and next generation sequencing technology and the dawn of like this metagenomic era, they discovered after SARS-CoV-1 emerged, they discovered HKU1 and NL63 shortly thereafter as causes of the common cold. And people don't really know the origin of HKU1. NL63 is believed to have come from bats at some point in time, maybe in like the 1800s, I think, doing like a molecular clock kind of analysis. So, you know, basically, there's probably other coronaviruses that are circulating and we just don't know about them. They're probably jumping into people all the time.
Hmm.
And if they don't cause severe disease or incite an outbreak or an epidemic, you may not hear of them or they might not become a thing. So it's only, you know, when you have emergence and sustained human-to-human transmission that you have these new viruses come out and become what, you know, what's happening with COVID-19. So
Yeah.
it's just, I think it's part of the personality of coronavirus to emerge like this. And there's no reason to think that won't happen in the future or continue to happen in the future.
Well, I think we're all worried that not only is it going to continue to happen, but it's going to accelerate. I mean, you know, this is a pretty compressed timeline to have SARS-1, MERS, SARS-2, you know. it's
Right.
the delta T is getting pretty small between these things.
It's
So,
all in the time of my training.
yeah,
Like,
yeah. I mean, and then of course, when we've got non-coronas like Nipah, you know, other horrors that are coming out.
right. I see Sarada has a question.
Yeah, yeah. Okay. Yeah. What else would you drug?
Yeah, so when looking at the coronavirus genome, the first two-thirds encode replicase proteins. There's 16. A lot of those are fairly conserved, like the RNA-dependent RNA polymerase. So chances are, the coronavirus encode 2 proteases. If you develop a protease inhibitor for one, it's likely that it might work against another one. So I'd say a lot of the enzymatic machinery that the virus leans on for replication are potential broad spectrum targets for antiviral drug development. Um, uh, the last third of the genome usually comprise basically the structural genes that decorate the outside surface of the particle, and then accessory ORFs that encode like interferon antagonists and stuff like that. And those accessory ORFs vary from coronavirus to coronavirus. So that's probably not a good broad spectrum target. And the structural genes like spike, which is the main determinant of protective immunity, where all your antibodies are raised to, that's pretty variable. So I think developing like a universal coronavirus vaccine or a broad spectrum immunotherapeutic is going to be hard.
Well, right. I mean, personally, I think we need both. I mean, obviously, if we had a broad spectrum one that's going to just wipe out anything that comes along, that'd be amazing. But I think at this point, you could get a $1 trillion check for one that just wiped out SARS-2.
Right.
So,
Yep.
let's see. We got another. So I will just tell the other the IT people that I can't. I'm not seeing any questions on the side, and I'm not seeing the ability to ask one. So I wonder if people got blocked in asking questions.
There's another question from Sarada, I see.
Yeah.
Can we design drugs that evade the proofreading enzyme? So that's a really interesting question. And Mark Dennison's lab has done a bunch of work on this. So there are nucleoside analog mutagens like 5-fluorouracil and stuff like that. Basically, the exonuclease cannot deal with that kind of molecule. But for some reason, no, it can. Maybe I'm getting this reversed. Basically, remdesivir has found a way to kind of evade the exonuclease activity, right?
Yeah, I heard that. Yeah.
Yeah, and one thing, it's hard to make, like basically you cannot knock out exonuclease activity in MERS, but you can in SARS. So really what you want to do in these kinds of studies is with reverse genetics, knock that out, have wild type, and then do drug assays to see, to pick apart the susceptibility or resistance to that kind of activity. Oh, there's more questions here.
Yeah, well, how about you? I'm going to pitch those to you, and you just say smart things.
Shoot.
Okay. Is there a physiological genetic switch that flipped with SARS-1 that altered the fatality of coronavirus sickness? Yeah. So, yeah. But what happened? Yeah. How did we go from just common cold to a super deadly coronavirus?
I don't think we know that. That is a great question. And it's hard to pick apart because these viruses are so big. And the common cold coronaviruses, some of them at least are quite different than SARS-1, SARS-2, and MERS. So trying to pick apart like the molecular determinants of pathogenesis is complicated. Um, but, you know, even for one virus, it's hard. You know, as we make mouse-adapted viruses, you know, you pass these viruses in mice, they acquire mutations that make them better and more pathogenic in a new host. And even determining which changes, even if it's 6 changes, you know, which changes might be driving that. It's hard sometimes to pick that apart, even in like a simple model like that. That's a good question. I don't know the answer.
Yeah.
So much for saying smart things.
Well, no, you're doing great. We got a question about receptors here.
Yep.
So SARS-1 and SARS-2 use ACE2. You know, MERS uses DPP4.
Yep.
Do you have an opinion on this? Do you think the virus just goes for something that pokes out and has a nice binding surface, or you know, is it cleverer than that? And you know, is there a reason that it would want to mess with our, for instance, angiotensin signaling?
Right. That is a great question. And, you know, there's some early papers on NL63, one of the common cold coronaviruses, saying that it also uses ACE2. So why 3 genetically disparate coronaviruses would want to use the same receptor is a good question. I think, you know, these viruses and the lives that they lead prior to emergence into humans is also like an interesting question, and may help kind of answer this question. You know, what these bat coronaviruses, what are they doing in a bat who don't appear sick, yet are harboring lots of different coronaviruses, sometimes at the same time? And but I don't know if they would, if there's a conscious reason to be potentially modulating the angiotensin system through the use of that as a receptor, or if it's just a bystander effect.
Yeah, I mean, as a protein engineer, I'm going to guess that it's probably just kind of random, but you never know.
Yep.
Oh, let's see. We've got. What do you think about co-receptors for SARS-2? I mean, we've got a list of a handful from SARS-1.
Yeah.
How hard were those to work out, do you know? And do we have any leads for SARS-2?
Yeah, and I think one thing to kind of differentiate here is, you know, a bona fide entry receptor versus something that might be an attachment receptor, or kind of help make the virus sticky to the surface of the cell as it's looking for something like ACE2. So I don't think it was that hard to discover ACE2. I remember the paper clearly when it came out when I was in grad school. But I think it's unclear if there are going to be bona fide co-receptors similar to like the HIV model, where there is CD4 and then co-receptors that help that virus enter cells. But, you know, there might be, besides receptor differences, but also protease differences, as the virus is gaining entry and the spike is getting cleaved to facilitate fusion. Are there differences in protease expression along your airway or from person to person? And there's definitely evidence that different coronaviruses utilize different proteases for that, that cleavage event, which is important for virus entry.
Yeah.
That's a good question, though.
I mean, so do you think it's unambiguous that SARS-1 is using, you know, so I made a list of things annotated as co-receptors, the DC sign integrin and lymph node C-type lectin. Do you know, are those quantified, and are they?
Oh, I'm sure of the ACE2 part of that biology. I'm less sure of DC sign. Like when I was in grad school, DC sign was a really hot molecule for different viruses for their entry, including like flavivirus and stuff like that. So, but I don't know. I don't really remember that data very clearly, and I'm not answering your question.
No, you did great. Let's see. Next up, we got a question from Michael. Is anyone considering, pre-exposure prophylaxis with some antivirals?
Not. I mean, I would think so, maybe eventually, but maybe not with the ones that I'm familiar with. I guess that would be driven by the safety profile and the route of administration for a given antiviral. If something was super safe and you could safely take it at home prophylactically once a week or every other day, I could see that being a thing. I just don't have a molecule that comes to mind where that would be the case.
Well, so what's your take on the, again, this is rumorology mostly with a little data to back it up, the Pepsid candidate. Have you heard this?
What, cathepsin?
No, Pepsid AC.
No. Like the heartburn thing?
Yeah, exactly. The heartburn. So, apparently, so in the initial Wuhan patients, that was the drug that most clearly gave signal between survivors and non-survivors. They had been having heartburn and were taking large amounts of Pepsid.
Okay.
And then when we had Kayvon Shokat and Nevin Krogan on, they had shown cell culture data showing that it could reduce titer.
Huh.
It's not clear. People think it might be hitting the RDRP. It's not really clear yet. Or it might be affect some sort of pH dependent, viral fusion, like endosomal escape or something.
Yeah, that's interesting. I had not heard that, but it makes me think that I'm trying too hard.
Yeah, we all just need to take a bunch of.
You can just go to CVS and get an armful of over-the-counter drugs, and I think that's it.
Let's see. Okay, here's a question from Adam. Does remdesivir affect transcription in human cells? Are there what sort of side effects do we need to be considering from this?
Yeah. So I've only seen like the preclinical data that Gilead has generated on this front, and they've done all the usual things that the FDA requires, mitochondrial toxicity and like measuring the effect on host RNA polymerases and stuff like that. And at least at the levels that you're giving this drug to people, that doesn't seem to be a major concern. Though there are, you do hear about ALT flare and liver tox with remdesivir in some people in these clinical trials, and other side effects. So I don't really know what's guiding that, other than it's thought to be reversible. But specifically on a molecular level, I don't really know what's driving that those adverse events. Good question.
Excellent. Let's see. Oh, here's a question about XON.
Yep.
Britt Glonsinger in the first episode of this series talked about this as well, that XON has this nasty habit of maybe coming back and knocking the drugs off. So it might be inhibiting remdesivir, et cetera. Question is, can you maybe make a prodrug strategy where you could have it where it wouldn't be knocked off and then would get activated and then go inhibit?
That's interesting. Yeah, I guess you could do that. Also on the table is, people are thinking about developing exonuclease inhibitors. So basically just targeting the enzymatic activity of XON. But yeah, I don't think people really know why XON is able to cleave off some nucleoside analogs that aren't supposed to be there, and others, it can't really do that. But Mark Dennison at Vanderbilt, that this is his bread and butter. And these are the kinds of things that he's interested in. And basically, can we use drugs like remdesivir or 2801 or whatever as tools to better understand the function of XON? But that's not really my focus, but that seems like a good idea.
Right. But I mean, in your wheelhouse would be, to me, it seems like if we're going to beat this thing, we're probably going to end up with some sort of drug cocktail,
Yes.
so you might take remdesivir and an XON inhibitor and, a fusion inhibitor and just throw the kitchen sink at it.
Yep. No, I completely agree. And like the analogy would be, the success of highly active antiviral therapy or HART, like we need to take a polypharmacologic approach to really combat this virus in the best way that we can. And, I think, you might also have some potential synergy, even if it's an additive effect. So like if remdesivir is a dud, if you put remdesivir with an immunomodulator or an IL-6 inhibitor or something like that, then all of a sudden maybe this dud is something that's actually much more promising.
Yeah.
So no, that's a great idea.
Yeah. Hey, we've kept you over. Do you have time for a couple more questions maybe, or should we let you go?
No, I've got another couple minutes.
Okay. Let's see. Do you want to speak to the male female risk ratio? Do you have any insights? We've heard a couple hypotheses so far in this series. Do you have a dog in the hunt?
No, unfortunately. One thing, I don't know if the other people who've spoken in this venue have addressed this, but so much information comes out every day that, I've been focusing on antivirals for coronavirus for the past 5 years. Just keeping up with that is not possible. So keeping up with all the different aspects of the biology that are revealed every day, I am at a loss for trying to balance that, do my job, and teach my kids how to read.
Yeah.
It's really hard. So, but it would be great if someone could enlighten me on the sex differences, but I can't really comment on that.
Yeah, one of the most compelling hypotheses we heard so far was a role for progesterone. That progesterone itself was, I can't remember the pathway that it was affecting, but it seemed to have an antiviral effect.
Yeah, there is some really cool data in flu showing differences between women and men in pathogenesis. But I don't know about this coronavirus.
Yeah, so yeah, I know I've tossed you a lot of things outside your realm of expertise, and do it again. Do you want to say anything about blood clotting?
Interesting. I wish that my colleagues were here because one of my colleagues here, Lisa Gilinsky, she studied some pathways related to clotting with SARS-1 in mice. And so this is a research area that she's looking to follow up with SARS-2 in our mouse models. Other than what I just said, I can't really elaborate because it's not really my research area, but that's definitely something that's on our radar, like collectively as a group.
Excellent.
Tim, a quick question. If you were thinking about a combination, a drug cocktail of remdesivir and an immune suppressant drug, do you anticipate any cross reaction between these two drugs or a combined side effect?
I guess that's always possible. I guess it would depend on what the combination is. You know, one thing that was a problem for SARS-1 and MERS was the use of immune suppressive drugs in the absence of direct-acting antiviral therapy. Mm. So basically, you know, do you really want to be carpet bombing someone's immune system when they have an ongoing severe respiratory infection? So, like, I think that coupling an immunomodulator in the face of pressure from a direct-acting antiviral may work better than each one separately. But I guess, like, you know, how they behave in a person and what adverse events might occur, I guess, would be should be taken on a case-by-case basis. But like, you look at what's happening even in the remdesivir trials, people are getting all sorts of stuff. Like, it's like Wild West combination therapy where they're just like, people are getting whatever. So it's really hard to wrap your mind around that. But, you know.
I think I saw a combination therapy where they were administering remdesivir in combination with, I forget, trying to look that up, a drug for rheumatoid arthritis. I don't know. Maybe, yeah.
I had not seen that.
Okay, one more question here from Wyatt.
Yeah, what do you think about losartan?
Interesting. So like these ACE or ACE2 inhibitors, this was something that a project that I tried to get off the ground in grad school for SARS-1 in mouse models. And I got some knockout mice. I tried to do some drug studies with losartan and SARS-1 mouse models. And I got inconclusive results. And then I went and did my postdoc, and no one ever followed up on it. But, you know, this was a question that was pitched to me on Twitter, like in January, like, will this have any effect? I don't know, it's hard to say. But, you know, these are obviously drugs that are capable of modulating these pathways in a person. People take these drugs all the time, they're safe. Whether or not they're going to have any effect on the outcome of infection, that remains to be seen. But that's a good question. I don't have a good answer. I could show you my lab notebook from like 2006 if you want, but we don't want to go there.
Yeah, and then something exciting that I've seen is the notion of administering soluble ACE2 as a sponge.
Yeah.
So that's really clever.
So yeah, and that so that work kind of spun out of SARS-1, and then so there's a guy in Austria. His name is Josef Penninger, and he's like an acute respiratory distress syndrome guy, and so he developed a soluble ACE2 inhibitor, I think originally for SARS-1, but they realized that if you can give this in models of acute respiratory distress, you can diminish the symptomology of ARDS by giving soluble ACE2. And so this is something that they were doing for H5N1 flu and for flu infections. And so I can totally see that. That's like a two birds with one stone.
Yeah,
kind of thing,
I like that.
You could be not only sopping up the soluble virus with this soluble receptor, but you could also be you know, diminishing the severe reactions that your respiratory tract might be having.
Yeah, I mean, but that observation, you know, coming back to one of the first questions, that makes me wonder if the virus chose ACE2 for a reason. You know, maybe knock out the anti-aircraft radar on your way in.
Right. That's a great question.
Yeah. Sarada, do you have one last one, or?
Sure.
Should we let Tim go?
I think, there's this question on, wait, let's see, this is from Hui.
Oh, okay. What do you think about the importance of maintaining weight during disease progression?
Like body weight?
Yeah.
In a person or in a mouse?
That was a question from Hui.
Yeah, I don't know.
Yeah, I don't really know how to answer that, but we do see some interesting things. So if in SARS-1 models, if you vary host genetics or you infect mice or different knockout mice, you basically have equivalent levels of replication. Some mice lose weight and some don't. Some mice basically get severe disease and some don't. So you have this like uncoupling of virus replication and weight loss and disease signs. But I'm not sure that I really understand what that question.
Yeah, I guess she said in disease prognosis. So.
Yeah, I mean, I know that like anorexia was a clinical sign of SARS-1 infection and was associated with disease.
Mm-hmm.
I haven't been reading any papers, so or I've been only reading a few. So I don't really have a good handle on all the symptomology in SARS-2 patients and if weight loss is associated with poor prognosis. But I guess fluid replacement and being in the hospital might help with that.
Yeah. All right, Tim. Well, that was amazing. Before you go, I just have to ask, bad religion or dead Kennedys?
Oh, dead Kennedys all the way.
See, I knew you were going to say smart things.
Coast to coast, man.
All right, Tim.
Yeah, thank you so much.
Yeah, keep doing the good work.
Thanks for having me. This was a lot of fun.
All right, dude. Stay safe.
Talk to you later.
All right. Bye now.